Well, let's get started. Good afternoon, everyone. Welcome back to the Kafner Research Council webinar series. As you may recall, we started off the CRC webinar series as an in-person coffee and conversation just before the pandemic. Dr. George Smith, our own Nobel laureate, was the inaugural speaker. Well, we changed our format to a webinar following campus shutdown back in the spring of 2020. We really would like to bring the coffee and conversation format back, but we'll wait until spring to do so. Today is our third webinar of the fall semester, and we have one of our newest faculty members, Dr. Craig Schenck, from the Division of Biochemistry, as our speaker. Dr. Schenck is a professor from the same division and a member of the Kafner Research Council, will make the formal introduction. But before she does so, I would like to say a few words about the Kafner Research Council. We formalized the CRC about 3 years ago as an advisory council that would provide advice to my office, that is the Kafner Office of Research. We do have faculty from every division in Kafner represented on the council. We also have staff, graduate students, and postdocs representation. This webinar is an example of some of the activities of the council, in addition to providing advice to me. They also organize the annual Kafner Research Day, help make important decisions regarding several research-related awards in Kafner, both faculty and student awards, and will also play a role in the new seed funding program we launched a couple of months ago called the Joy of Discovery Seed Grant Program. When they help make these decisions in the Office of Research, they are representing you. In other words, the Kafner Research Council plays an important role in shared governance in our college. Before I hand over to Antje, let me remind everyone that you need to post your questions in the Q&A box or in the chat room. Dr. Hayes will be moderating the Q&A session in the end. So without further ado, Antje, the floor is yours.
Thanks, Shibo. So it is my great pleasure to introduce Craig Schenck, who joined our department, the Kafner side of the Division of Biochemistry, in the summer, so earlier this summer, 2021, and is actually my neighbor now on the same floor. So this is great. So Craig actually started his career in plant biology, and with a minor in biochemistry from Ohio University. He received his bachelor degrees and then also his master degrees from the molecular and cellular biology at Ohio University, and we will not hold that against him. But he then moved on to perform his PhD in the botany department in Dr. Maeda's lab, performing or studying amino acid biosynthesis and metabolism. And after a very successful PhD, he then moved on to join Rob Last's group at the University of at Michigan State University, and where he then moved on to study metabolic diversification and look more at the evolutionary aspect of diversification of different metabolisms in plants. And this is now what he actually will tell us also about, and he what his research program will actually entail here. But before he does that, before I hand this over to Craig, I want to actually make two notable honors that Craig actually received. So he is a recipient during his postdoctoral work in Rob Last's lab. He received the NSF postdoctoral fellowship, which is very difficult to obtain. It's very competitive. And he has been very active in our American Society of Plant Biology, and he received the highest honor in 2019 by the Society, by ASPB, and received the Eric Cohn Young Investigator Award. And with that, Craig, I let you talk about your exploration into evolution of plant chemical diversity using acyl sugars. Thanks.
Thank you so much for the introduction. Trying to share my screen right now. How's that look? Okay, great. Well, Antje, thank you so much for the introduction, and also to Shibu and the Kafner Research Council for providing me this venue today to share my work. So as Antje said, I've recently started just this past summer. So most of the work I'm going to talk to you today is work that I've done in my postdoc. But much of what the lab will focus on in the future will be somewhat of a continuation of some of these projects, but also some newer ones. So what I'm really going to talk today about is how plant chemical diversity has evolved, and what are the impacts and implications of plant chemical diversity, not only for plants, but also for humans. Whoops. Let me turn on my laser pointer really quickly. Okay, so plants are really the best chemists on Earth. I don't think there could be any other argument. And they have to be, because they're rooted in place and they can't rapidly escape when harsh conditions arise. So they've evolved a diverse chemical arsenal in order to enable environmental interactions. And these plant chemicals really serve as the language of life. Some of these chemicals enable positive interactions, for example, pollinator attraction, so over here. But then there's also a set of chemicals that is involved in negative interactions. For example, deterring herbivores and things like that. So in addition to their roles in plants, though, they've also been co-opted by humans, where we're familiar with their nutritional role, but they also have roles in industrial applications. And most importantly, they're involved in diverse medical applications. So for example, this is a compound over here that's called Artemisinin. It's derived from a medicinal plant called Artemisia annua, and it is an anti-malarial drug. So this is just one example of a medicinal application of plant metabolites. But what I'm particularly interested in is how this chemical diversity has emerged in plants, and what are the alterations at the biochemical level or pathway level that have enabled this extreme diversity in chemistry. And so I use the rapidly evolving plant specialized metabolic pathways as a model to investigate these sort of questions. So what are plant specialized metabolites? These are compounds that I was just alluding to, but they are typically biologically active end products. So some examples are shown here as well as examples that were shown on the last slide. Another aspect is they're often produced in limited plant species. So unlike core metabolites like amino acids or nucleotides that are found in all plants and even bacteria and things like that, specialized metabolites are restricted in their phylogenetic distribution. So one nice example are capsaicinoids, which are the spicy compounds in peppers. These, of course, are restricted to only pepper plants and not closely related species. So that's an example of that. So the last characteristic is that these specialized metabolites are produced in a cell or tissue specific manner. And another example of that is artemisinin and other terpenoids. These are produced in specialized cell types called trichomes, which are hair-like structures on leaf and stem surfaces. And I'm going to talk a lot about these moving forward. It's their lineage specificity and their tissue specificity, though, that used to hinder the discovery of specialized metabolic pathways. But now with the advent of transcriptomics and very cheap sequencing, comparative omics approaches are elucidating biosynthetic pathways in plants because of the lineage and tissue specificity. So this is kind of facilitating pathway discovery. And so one major question, though, is where has this chemical diversity come from? And can we harness the power of these pathways and the enzymes to enhance crop resilience, or use them in synthetic biology platforms to enhance production of medicinal plant compounds? So we do know that the diversity is mainly derived from core metabolism. So specialized metabolites are derived from core metabolic pathways like amino acids, nucleotides, fatty acids, and so on. These pathways are highly conserved in plants, and they provide the precursors to specialized metabolites through lineage-specific metabolic pathways. But really, although I've drawn lots of arrows, we don't know in many cases what the metabolic pathways that are connecting certain amino acids to downstream specialized metabolites. And we don't really understand the mechanisms that enable this chemical diversity in plants. And so I'm going to show you some stories that are kind of working at the interface here between core metabolism and specialized metabolism, and highlight some mechanisms that contribute to plant chemical diversity. And our hopes is that if we can better understand metabolic pathways and identify enzymes with unique activities, that we can use these to enhance biologically relevant plant metabolites. So the specialized metabolic pathway or class of metabolites that I've worked most on are acyl sugars. And I'll explain to you why these represent such a good model system to kind of explore evolution of plant chemical diversity. So this is what they look like over here. They have a sugar core, and it can be a disaccharide or a monosaccharide. And they have carbon chains that decorate the acyl sugar. These are found across the Solanaceae family. This is a family that contains species like tomato, potato, peppers, tobacco, petunia. So lots of agriculturally, but also horticulturally relevant crops. Acyl sugars are generally found across all of these species, though their structures can be quite varied. Another aspect, which is advantageous, is that they're produced in trichomes. And I mentioned these before, but the fact that they're made in trichomes enables pathway discovery quite readily. The other thing is that these acyl sugars are also involved in plant defense. So either in direct defense or have also been implicated in the indirect tritrophic interaction. So a bit more about the biosynthesis of acyl sugars, because that's what I'm going to be talking most about. Like other specialized metabolites, these are derived from simple building blocks from core metabolism. So you have sugars and CoAs, in this case, derived from branch chain amino acids. The condensation of these two substrates is catalyzed by a group of enzymes called acyl sugar acyl transferases, which add the acyl chains onto the hydroxyls of these sugar cores. And it's the elaboration of this core metabolism and the diversity of acyl-CoAs that can then lead to lots of different acyl sugar structural variants. So you can have more than just these 4 and 5 carbon acyl chains. So you can have longer ones, you can have shorter ones, you can have diverse branching patterns, and this leads to a diversity in acyl sugar variants found within a species or across a genus or even the entire family. There's a tremendous amount of acyl sugar diversity. So it's really the trichome accumulation and biosynthesis together with diverse products within a species and across the family that then make acyl sugars really a good model to explore the evolution of specialized metabolism. So I'm going to tell you 3 stories today about how different plant species have kind of elaborated on these mechanisms to enhance the acyl sugar diversity. One that kind of fits within this area, connecting this core metabolism with specialized metabolism. Another that kind of targets the last step in the biosynthetic pathway. And then another, actually how the pathway has relocalized to another tissue, resulting in some chemical diversity. And so I hope to tell 3 vignettes, all highlighting mechanisms that are enabling chemical diversity. So we're first going to start by a gene co-option event from core metabolism, and this is work that we've done in tomato. And so I already mentioned the biosynthetic part of this work, but it's good to keep this in mind still. But one of the first things that I did during my postdoc was actually screen diverse Solanaceae lineages and looked for acyl sugars across those. And then what I did was I mapped acyl sugar diversity. In this case, I actually looked at the acyl chain lengths. So these acyl chains that are attached to the sugar core itself. And then I mapped those onto a phylogeny. And what I found is that many species made acyl sugars with quite diverse acyl chain lengths, but only some species produced really long acyl chains, 12 and 13 carbons long, whereas others produced only acyl chains up to 8 or 6 carbons long. And these were phylogenetically restricted. And one thing that highlights is that you're getting more than just 4 and 5 carbon acyl chains, so there must be some sort of elongation mechanism of these acyl chains. So I wanted to investigate this elongation mechanism. And another interesting finding is that precursor feeding studies that had been done, I don't know, 20 years ago or so, had indicated that a few of the plants that make long chain acyl sugars actually use a 2-carbon elongation mechanism that would be analogous to fatty acid biosynthesis. So that's where my head went. And I wanted to pursue this hypothesis. So do these acyl chains get elongated by a fatty acid biosynthetic-like pathway? So just a little bit of background about fatty acid biosynthesis, and I'll try and keep this brief. Fatty acid biosynthesis, the first step is catalyzed by this condensation reaction, an enzyme called beta-ketoacyl ACP synthase. I'll just refer to it as Cas enzymes. So this condenses acetyl-CoA with malonyl ACP to make a 4-carbon product. This can be further elongated, and I'm leaving some steps out, but they're not so important for today. This 4-carbon intermediate can then be condensed again with malonyl ACP to make a 6-carbon product. So this is that 2-carbon elongation. So this can happen quite a bit, catalyzed by a different enzyme, a Cas1 enzyme. And then there's a final step in the elongation pathway, at least in Arabidopsis, where a 16-carbon long intermediate can be elongated to an 18-carbon long product, and that's catalyzed by a Cas2 enzyme. So this is what canonical fatty acid biosynthesis looks like. So I thought that perhaps something similar was going on in tomato, but that it was using acyl sugar substrates. So it's possible that this kind of looks very similar, except that instead of acetyl-CoA, you have a 4-carbon branched CoA as a starting point. And this could make a 6-carbon acyl chain. You could also elongate that to an 8-carbon, a 10-carbon, or a 12-carbon long acyl chain. So this is kind of the hypothesis that I was trying to test. And so this seemed like a useful or a reasonable hypothesis. So I used comparative genomics and transcriptomics, and then I also coupled that with making knockout mutants using CRISPR-Cas9. And so when I looked for homologous Cas enzymes across the Solanaceae family, what I find is 3 clades. And they're highlighted in these gray boxes. And each of these clades contained one of the Arabidopsis sequences, highlighted here. But in each of these gray groups, there were at least 2 sequences from every Solanaceae species that I had analyzed. So what this indicated was there was a gene duplication event, at least at the base of the Solanaceae. And within each of these gray groups, there was another group of sequences that were highly enriched for Solanaceae sequences. And then I next wanted to look to see if any of these genes were highly enriched in the trichomes where acyl sugar biosynthesis is occurring. That would indicate maybe their involvement. And so that's what I'm showing here, where I'm showing trichome over stem or leaf expression. So high values here indicate trichome enrichment. And so each of those red groupings contained Cas enzymes that were highly enriched in the trichomes. To me, this suggested that these might be involved in acyl sugar biosynthesis. So the next thing I did was I created knockout mutants using CRISPR-Cas9 for each of the tomato sequences that fell within these red groupings. And so this is, I'm only going to show data for 2 of those mutants today because they're all quite similar in phenotype, at least chemical phenotype. So this is an example of a trichome enriched Cas1 enzyme. So I was able to get 2 independent mutations here, or 2 independent alleles. And then we looked at the chemical phenotype of these. So we screened acyl sugars from our individual CRISPR mutants as well as wild type. And one thing we saw here, so each group of bars indicates a type of acyl sugar. These acyl sugars contain only short chains, so 4 and 5 carbons, and these are ones that require no elongation. These were not any different between wild type and mutants. However, those acyl sugars that contain long chains, like a 10 carbon or a 12 carbon acyl chain, those that do require elongation, those acyl sugars were all reduced in the mutants compared to wild type. And so this suggested their function in acyl sugar biosynthesis and elongating these acyl chains. And since this Cas enzyme is closely related to the canonical fatty acid biosynthetic gene, we also looked into the fatty acid profiles just to make sure that this gene wasn't also involved in canonical fatty acid biosynthesis. And the mutants were not significantly different in any fatty acid that we could detect. And this is data for another of those CRISPR mutants. This time we were targeting the Cas2 enzyme. That's down here, so it's highly enriched in the trichomes. This one had a similar chemical phenotype. It was slightly different. Again, acyl chains that require no elongation, those acyl sugars were unaffected, but those that contained a long acyl chain, either 10 or 12 carbons, were affected, but in a different way. We saw a redistribution here instead of a total reduction in long acyl chains. So in the mutant, we see a reduction in acyl sugars that contained a 12 carbon acyl chain, but we actually saw increase of acyl sugars that had a 10 carbon acyl chain. So there was some sort of redistribution here. We also confirmed this result using GCMS, which is a different analytical approach, but we got a consistent result. And I should have shown a picture of the mutants in the previous slide, but this is what the mutant looked like compared to the wild type. No major whole plant phenotypes. And so, although we didn't look at the trichome phenotype, we would like to look at the phenotype of the trichomes in these mutants. We are also interested in understanding how this co-option event has occurred at the molecular level. So the next step really is to do in vitro biochemistry and understand how at the structural level or at the active site, how these enzymes can accommodate slightly different substrates than what would typically be used in canonical fatty acid biosynthesis. And so those are the things that we're currently pursuing. Okay, so that's story number one, showing how a gene from fatty acid biosynthesis was co-opted into specialized metabolism and how this contributes to acyl sugar diversity. So the next vignette is about pathway discovery in a different group of plants, this time Nicotiana. So this is the tobacco genus, and how enzyme promiscuity or the maintenance of enzyme promiscuity enables chemical diversity. So this project started actually with chemical screening acyl sugar diversity across the entire Nicotiana genus. And I won't get into details here, but we extracted acyl sugars from leaf surfaces and used a couple different analytical platforms to get a sense for the structural diversity of these acyl sugars. And this is work that an undergraduate, a summer REU student actually did with me while at Michigan State, Mackenzie. And we ended up screening close to 45 species. And I'll just give you a sense of kind of what the data looks like if it comes up. So these are 3 different or representative species that we screened. These are LC chromatograms where we've separated acyl sugars. You can generally assume that every peak here corresponds to an acyl sugar. I'm only showing a portion of this chromatogram. And you could count up the acyl sugars, and kind of every peak here is, if it has a different retention time, is probably a different structural variant. And then you can look in these peaks and actually look at the fragmentation pattern to get more information about the structures of these compounds. And if you do this across 45 species, you can imagine that you start to get a lot of structural information. And we struggled with how to kind of make sense of this. But what I thought we should try and do is place structural diversity again into a phylogenetic context. So this is something that I do quite often. So what we did for this project, we took all the species that we screened. We looked at the acyl chain types. And again, we mapped that onto a Nicotiana phylogeny to see if we could pick out any trends that we might want to further investigate. And so red boxes indicate that plant species produced acyl chains of those lengths, and a white box indicates absence. So a couple things to note is, first, we didn't observe acyl chain lengths greater than 8 carbons. And I think this has a lot to do with the acyl chain elongation mechanism that could be in play here. And it's likely different than what I just showed in tomatoes. But the other things to note are 5 carbon long acyl chains were almost uniformly distributed across the entire genus. The longer CoAs were a bit more sporadically distributed. And the last thing that I was most interested in is there was one acyl chain type that was restricted in its into a few closely related species. So you can see that only a single clade contains this particular acyl chain type. And I'm calling it 5D here because it was 2 mass units less than a typical 5 carbon long acyl chain. So it indicated there was a desaturation there. So we wanted to look a little more into these particular species. And so we used GCMS to kind of decipher the structure. This provides a bit more information about the structure of the acyl chains themselves. And what we found is that 5D acyl chain, in fact, was a 5 carbon long acyl chain with a desaturation called Tiglyl-CoA. And in Nicotiana acuminata, this accumulated up to 75% of all the acyl chains on those acyl sugars. So an uncommon acyl chain, and it accumulated quite a bit. So we wanted to resolve these structures more accurately. So we used NMR, and we resolved the structures of 2 tiglated acyl sugars, one that has 4 acyl chains, another that is almost identical, except that it has an additional acetyl, a C2 acyl chain here. And this was work done by Talani Anthony, who performed all the NMR experiments. And so given the unique acyl chain and the accumulation of that acyl chain, and also I didn't mention, but Nicotiana acuminata also produced about 20 or so different acyl sugar structural variants, which is quite a bit. We wanted to do pathway discovery to understand how it can do this. And so we could take advantage of the trichome specific synthesis of acyl sugars, and we used a transcriptomics enabled approach for pathway discovery. And so what we did was you scrape trichomes and then extract RNA from those trichomes and compare that to the underlying stem or leaf tissue. And then you can do a de novo assembly using Trinity, and then you have your transcriptome. Then we can take advantage of the fact that this pathway has been characterized in tomato, and we used a comparative genomics approach using characterized tomato ASATs to look in the Nicotiana acuminata transcriptome for homologous enzymes that were also highly enriched in the trichomes. And so we ended up finding 4 candidate genes that kind of fit our criteria. The next step was to validate these in vitro, and also in vivo. So I'm not going to talk about the in vivo validation, but I will talk about the in vitro biochemistry of these ASATs. So I started with the first step that seems like a logical place to start. And also it helps that these substrates are available from Sigma. So it's a pretty easy enzyme to assay, but you take your purified enzyme, you add substrates, you incubate it for a bit, and then you monitor the reaction for a monoacylated product using LCMS. And so that's what we did, and that's what we see. So we see a monoacylated product, a sucrose with a 1 acyl chain that's present in this enzyme assay that's not present in the negative control, suggesting that this catalyzes the first step in the reaction. We could then go on and do kinetic analysis, and this just demonstrates that this enzyme behaves similarly to the previously characterized ones from tomato. So given the extreme structural diversity in acyl chain types, we also wanted to investigate whether or not this ASAT1 could use more than just this vanilla IC5-CoA. So we tested it with a broad range of acyl-CoA donors. And also we tested it with a range of acyl-CoA acceptors. So we swapped out IC5-CoA for others, and we also swapped out sucrose for other sugars. So the first thing is that using a diversity of acyl donors, including aromatic acyl-CoAs all the way up to long unbranched acyl-CoAs, in this case, a 14 carbon long CoA, this enzyme accepted them all. Although the degree of activity varied quite drastically, it was able to use all the substrates that we tested it with. So it displays extreme acyl donor promiscuity. However, when we we altered the sugar, it's much more specific. So it uses the disaccharide sucrose quite well. The only other disaccharide that it can use was tree hollows. However, this was about 50-fold less activity than with sucrose. And it didn't accept any other disaccharides. We also tested monosaccharides, and it had no activity. So this shows the promiscuity, but also some specificity in terms of the acyl acceptor. And so this is characterizing the first step. Then we wanted to characterize the rest of the steps. Characterizing the rest of the steps can be a bit trickier because oftentimes the pathway intermediates don't accumulate in plants. So the best way around this was to do in vitro pathway reconstruction. And so simply put, we took all our candidate enzymes and put them in a tube at once and used a couple different CoAs as well as sucrose, and then monitored those reactions for monoacylated products, but also acyl sugars with more acyl chains. And so when we did that, and I'll show the data kind of maybe in a more simpler way and then in a more complex way. But if we just add ASAP1, we only get production of a monoacylated product and not higher order acylated sucroses. If we add 1 and 2 at a time, we get both mono and diacylated products, as we kind of expected. When we add ASAT3, we start to observe triacylated products. And finally, when we add ASAT4 together with the first 3 steps, not only do we get mono, di, and tri, but we start to accumulate a tetraacylated sucrose. So this in vitro reconstruction demonstrates the ability of these enzymes to make acyl sugars in vitro. This is what the data actually looks like. So we get more than just 4 peaks or 4 products that are formed. In fact, we can actually make an acyl sugar with 5 acylations using these 4 enzymes. And we get multiple products that are diacylated and multiple products that are triacylated. And so this just kind of reiterates the metabolic capacity of these enzymes to make diverse products in vitro. And so this was really just the starting point. We've done a lot of additional experiments where we've varied the acyl CoAs, and I'll just show you one. But essentially what we wanted to do was push the system and see how much acyl sugar complexity we could produce in a single enzyme assay. And so and this is kind of taking advantage of the promiscuity of these enzymes. So in this experiment, we mixed diverse acyl CoAs together with sucrose, and we used our 4 candidate enzymes. And we were able to make monoacylated products and diacylated products, and diacylated products with 2 long chains instead of a short and a long chain. And so you can see this could go on, and I could show you a lot of the chromatograms, but just to summarize this and save the details, we could make greater than 25 acyl sugar variants in vitro, which is beyond the diversity that we can find in any Nicotiana plant extract. So this demonstrates the potential of using these sorts of enzymes in synthetic biology platforms to make diverse acyl sugar products, but potentially diverse other types of products with biological activities that are relevant. And so to summarize that's an example of how maintenance of enzyme promiscuity enables plant chemical diversity. And we hope that we can further kind of co-opt that promiscuity to make much more diverse products in the future that may have novel biological activities. Okay, so the last story that I'm gonna touch on today is actually a story at a gene expression level, and how gene expression changes have led to pathway relocalization. Okay, so this is work that I did in tomatillo. This is, I wouldn't necessarily call it a crop. People often refer to this colloquially as green tomato. It's not a tomato. It's closely related to tomato, but it's its own entity. This is a plant that's widely consumed in Mexican cuisines, and you may have had it in salsa verde. And maybe you've come across this in the grocery store. I'll go back here. This is a plant that, so this is what the fruit looks like, and it's surrounded by what's called a calyx. More commonly, this is referred to as a husk, which may be involved in protecting the fruit. But maybe you've seen it in the grocery store and with a dried out papery husk. But if you ever touch a tomatillo fruit, you may notice that they can be extremely sticky. And I wanted to know what the nature of this stickiness was, the chemical nature. And so I also thought, since it's closely related to tomato, I might as well screen the acyl sugars and see what's there. And I expected that I would find copious amounts in the leaf, but when I looked, you don't see that many. And in fact, you see abundant acyl sugars on the fruit surface. So likely those sticky compounds on the fruit surface are acyl sugars. And in fact, when you grow this in the mostly the greenhouse, where there's a little more pests around, sometimes you'll find flies and other things that are trapped to the fruit surface itself. But what was intriguing here is that we were detecting acyl sugars on the fruit surface, but not on the leaves. And so questions of how did this pathway move to the fruit surface from the trichomes started to arise, and we wanted to investigate that. And so one thing I did was I also noticed that sometimes when I looked at the fruit, there were glossy regions and very sticky regions, but there were also some regions of the fruit that were not sticky whatsoever. So I wanted to see if there is any distribution of these acyl sugars across the fruit surface. And so I screened acyl sugars from different zones of the fruit. I'm calling zone 1, 2, and 3. And at least for this extraction, I found acyl sugars that were highly enriched in zones 1 and 2, but not in zone 3. And so this led me to this working model where acyl sugars are produced and secreted perhaps kind of at the top of the fruit surface. And then as the fruit develops, or maybe as gravity kind of pulls the acyl sugars down, they can float down to kind of coat the entire fruit surface. But the problem with tomatillo is it's not a great model system. There's really no genetic resources. No genome was available. So it was hard to develop the tools to try and address the questions that I wanted to address. But luckily, almost simultaneously while I was screening these tomatillos, a closely related species called Physalis griseae, its genome was released. And it was a really high quality genome. So I thought, oh, I'll just screen Physalis griseae. It'll make acyl sugars on the fruit. And then I can identify pathway genes and things like that to answer some of these questions about how the pathway relocalized. Well, when I screened acyl sugars from Physalis griseae, I found the more expected outcome of acyl sugars on the leaf, and in this case, also the calyx. I will say that for this species, there are trichomes on the surface of the calyx. So the fact that acyl sugars were present there was not too surprising. But what was maybe disappointing, though not surprising, is the fruit actually contained no acyl sugars. So now I had a genetic system that I could work with, but one where it didn't make acyl sugars on the fruit, but maybe this provided a good comparison. So the next thing we wanted to do was trying to identify some of the pathway steps so we could address questions about how acyl sugar biosynthesis moved from trichomes to the fruit surface. And to do that, we went back to using comparative genomic approaches. In this case, we identified a handful of ASAT enzymes, which we validated in vitro. This is all, again, from Physalis griseae. So we used the two enzymes that I've highlighted here in red. And in vitro, we were able to make both mono and diacylated products, suggesting that these act consecutively and are involved in acyl sugar biosynthesis. And also, these provide the tools that we can now use to investigate how acyl sugar biosynthesis in tomatillo moved from the trichomes to the fruit surface. So now that we have the pathway enzymes, or at least two of them, we can use those to our advantage. And we even think that we may have identified a putative structure on the surface of tomatillo that could be involved in the production and secretion of acyl sugars on the fruit surface. And we did this using scanning electron microscopy. And so what you're looking at here is kind of the top of the fruit. So this area where the fruit is connected to the receptacle. And within this area, it was highly concentrated for structures that look a lot like this. And these structures were not found anywhere else on the fruit surface. And they do look a bit like stomata, which are involved in gas exchange. But I feel like if they were to be stomata, they might evenly be distributed across the fruit surface, yet they're only found at the extreme top of the fruit. And this kind of matches well with the hypothesis that acyl sugars may be biosynthesized and excreted somewhere at the top of the fruit, and then over time they kind of flow down and coat the fruit surface. So what we're currently trying to do is to identify if any of the ASAT genes that we've found are expressed in these sorts of regions. So we're tagging them with GFP and looking to see if ASAT expression can be found around these sorts of structures. And so that's why elucidating the biochemical pathway has given us tools to kind of investigate these interesting questions about how the pathway may have been relocalized to the fruit surface. And so the other thing that I'm interested in doing with this project is understanding when this trait emerged. So when did fruit localized acyl sugar biosynthesis emerge? So this gets back to what I like to do, is put things into a phylogenetic context. Here, we've only screened 3 different species, so you can't really take anything from this analysis quite yet, but this is where we're headed. So what we plan to do is screen across the Physalis genus, screen for acyl sugars on fruit and both leaf surfaces, and then map that into this phylogenetic context. And hopefully we can get some better resolution as we grow some of these plants and do chemical analysis. But you can imagine a scenario perhaps where this trait maybe emerged at the base of this genus and is found in these 2 species, but maybe this trait was lost somewhere after that. Or you can imagine a scenario where this trait evolved once, just in this very limited group of species. Or perhaps this trait could have evolved multiple times independently. And so we're hoping to pinpoint a timing for when this trait emerged, use the biochemical tools that we have to investigate how a metabolic pathway could move from a trichome to a fruit surface, and how this could be accommodated at a metabolic level, while also trying to learn some interesting things about the evolutionary processes of these events. And so to kind of summarize maybe all the vignettes, or to put a bow on everything, I hope that you've got from my talk, seeing how screening across diverse plant species, you can really learn about a lot about evolution, much more than you could by just looking at a single species alone. And that there are diverse biochemical mechanisms that enable plant chemical diversity, and we're just scratching the surface. And in the future, we plan to look not only at acyl sugar biosynthesis, but diverse metabolic pathways across plants to understand more of these evolutionary mechanisms, but also to kind of expand our toolkit, so that we can use plant enzymes to enhance crop resilience or in synthetic biology platforms. With that, I'd like to thank Rob Last and the Last Lab. Like I mentioned, most of this work was done during my postdoc work. I'd like to thank NSF for funding the work, and some collaborators who helped immensely. I'd also like to thank not only the University of Missouri, but 2 undergrads, Connor and Will, who are actively doing research in my lab now, who I didn't have time to talk about their work today, but hopefully in the future I can. And with that, I'd also like to thank all of you for your attention.
Oops. Well, thanks, Craig, for a very
You're
exciting
welcome.
and interesting talk. So
Should I
please,
stop sharing, or?
it's up to you, I think. Let me see whether if people have, anybody who listens has questions, please add them to the Q&A session, and I will read them out loud then. Or also the chat room. Let me just see here what, I think I saw one. I think Lloyd said tomatillos are great for, I actually cannot see this here.
Chili Verde, I think he says.
Yeah, so thanks, Lloyd. But I would like to then start off with a question here. So you had said initially that acyl sugars were used as an antimicrobial or defense mechanism because they're often in trichomes. So that makes sense because plants use trichomes as one part of their defense. So what do you then hypothesize happens in tomatillos or in this physalis when it moves actually more into the fruit and only part of the fruit, or at different portions of, I guess it was the husk?
It's a good question. Yeah, we do know that these possess antimicrobial activity and anti-herbivory activity. I think these are broadly defensive compounds. They're not targeting specialists. And so the fact that this pathway is moved to the fruit surface, and what are the biological implications of that in tomatillo, it's hard to say. We'd love to address that. So we're working on knocking out some of these pathway enzymes using CRISPR-Cas9 so that we can make non-sticky tomatillo fruits. We could then test these out in the field and see what happens. Tomatillo and other physalis make a lot of other very interesting defensive compounds called withanolides. So it's possible that, in some instances, the chemical arsenal is just co-evolving within a plant. And, as acyl sugars go away, maybe another compound steps to the front and is their primary defensive compound.
Yeah, let me just see. I'm just looking also at the chat. It's actually great presentation and TM, great presentation. Thank you. And I can't see actually who wrote that. But so let me just see here. Actually, I had a couple of questions. So you had said, so you use a lot of genomics and transcriptomics to basically place certain pathways or enzymes into pathways and to reconstruct. Is there any evidence that part of this acyl sugar condensation or elongation, these different lengths are also regulated more on a post-translational side, or is it all transcriptomics or on the transcript on mRNA level?
It's something we haven't looked at, but I do think it's a good system where we could do proteomics and kind of figure that out. I think that's one place this project could go forward. Just understanding, post-translationally how things are regulated, because we know that there are other specialized metabolic pathways that are post-translationally regulated. I think people just don't often look there and specialized metabolic pathways. I think we're at the point now where we're just doing a lot of pathway discovery, identifying the core sets of genes involved, and then, maybe we'll be seeing those sorts of analyses in the future.
Yeah. Well, let me see. I'm just looking through. Now, also said, thank you for a nice talk and nice and interesting talk. So, there is one longer one. Let me see. The diversity of specialized metabolism within a class is interesting. Do you think this just combinatorial chemistry, or could be something more? Thus, is there any evidence that certain forms of acyl sugars are active against specific microbes to generate broader defense profiles?
Yeah, that's a really good question, Lloyd. There is indication that particular acyl chain lengths or particular acyl sugar types do have enhanced biological activity against certain pests. And this has probably been, this is probably a main evolutionary driver for the complement of acyl sugars that you see in certain species. And perhaps why you see more limited acyl sugar types within a species, yet, when you take these enzymes and do in vitro biochemistry, you can produce a huge arsenal of these. So, I guess one thing is I still am not quite clear how plants are regulating the products that they're making, given the extreme promiscuity that I observe in vitro. But there are certainly examples of acyl sugar types having enhanced biological activities. So a plant must be able to regulate this in some way. And maybe there's enzymes involved in kind of not necessarily proofreading, but making sure that the proper or the optimal acyl sugars are being produced at a given time and location.
Mm-hmm. Yeah. So actually one part I'm very intrigued by this structure that you identified, being more a cell biologist, the structure that potentially secretes then these acyl sugars on the physalis. And I actually, so when you break off trichomes, it leaves this footprint behind that is often raised. Have you thought about whether that may be like an altered trichome?
It could be, yeah. I mean, I think these sorts of structures are also found on citrus fruits. I guess I forget the name of those structures, but these are the ones that are releasing the essential oils, I guess, of the on the fruit surface of oranges and lemons and limes. So yeah, this could be a modified trichome in some way. I'm very intrigued to continue investigating this problem. So any advice or suggestions are welcome for that.
Okay, and then I think There was one more. Could the regulation, the last question, and then I think we wrap up. Could the regulation be related to subcellular or cell-to-cell transport of specific forms of acyl sugar? And I think that was also from Lloyd.
Yeah, it's another good question, and something that we just don't know a whole lot about, the subcellular organization of this biochemical pathway. So there's a lot of moving parts. You know, you have CoAs coming from branched chain amino acid biosynthesis. We're implicating elongation mechanisms. You have your acyl sugar acyl transferases. There could be a lot of subcellular partitioning and regulation that's occurring here that we just, right now, we just don't know anything about that. So that's a good question in an area that really needs to be further investigated.
Mm-hmm. Well, yeah, thanks again for everybody who attended this seminar, and thanks for Craig for giving a great talk and for us to learn more about our new member of our faculty and Kafner. So let me just see here. I'm not sure whether Felix will have a final say. I actually can't see here, but I think he's actually, I don't see him anymore. So I will do the closing part. There will be more CRC webinars coming up in December. So please keep your eye out for future seminars. And we hope we see you then at other CRC webinars. So thanks everybody, and thanks Craig.
Thank
Bye.
you.